Hello, I'm Pamela Bursler, Executive Director of Pacific Horticulture Society, and I'd like to welcome you to the Digital Classroom, a series of discussions and interviews bringing compelling topics of environmental science into the realm of horticulture, gardening, and landscape. The first 5 topics in our series have been underwritten by the Pendleton and Elizabeth Carey Miller Charitable Foundation, and we thank them very much for their support. All of the talks that we've put together so far and the ones in the future will be found at pacifichorticulture.org/digitalclassroom. And if you have any questions or comments, want to reach us on anything really that we're talking about, email us at digitalclassroom@pachort.org. So today we're talking to 2 PhD candidates from Simard Lab at the University of British Columbia in Vancouver, Alan LaRocque and Katie McMahon. Alan and Katie are working with Suzanne Simard on her research into various aspects of forest ecology, and you will know her best as the person who pretty much explained to all of us that trees talk to each other and work with each other and even have relationships with each other. The discussion today, however, is going to focus on some of the fascinating underground cooperation between mycorrhizal networks and the forest, and how trees work to build and manipulate their own microbiome. We're going to start with Alan LaRocque, whose research into the complex symbiotic relationships between trees and mycorrhizal networks takes him to the salmon forests of British Columbia. Then we'll turn our attention to the research conducted by Katie McMahon into terrestrial reclamation and rebuilding of soil food webs after disturbance. Her work has been conducted primarily at mine sites, so it can't be nearly as bad as our gardens, right? Okay, let's start off with Alan. Alan, take it away.
Good morning, everyone. My name's Alan, and it's a pleasure to be here with you. I'm just going to jump straight into my presentation here. There, is that working, guys?
It is.
How about that? Awesome. So my presentation here is split into 2 little parts. I'm going to start by introducing the Simard Lab, which is quite a large group and diverse bunch of people. And then I'll move on a little bit to talk about my own research. So the Simard Lab is at the University of British Columbia in the forestry department, and it's one of 4 labs that are a part of what we call the below ground ecosystem group. And we study, those 4 labs study all different facets of soils and forests primarily, but we also look at estuaries, valleys, mountains, and the Simard Lab looks at fungi. So here's our captain, Dr. Suzanne Simard. So she's been professor at forest ecology at UBC for about 20 years now. And she came from a professional forester background, which kind of is quite rare to have academics who also have kind of inside knowledge of industry and government processes. So that's been really helpful for me personally, getting to know her. And her academic work has been on below ground mycorrhizal networks, which we'll get into in a moment, complexity science, and especially resilience and adaptive management in forestry, cities, and other settings. If you want to learn more about her, I put a bunch of links here to the lab, the Mother Tree Project, which is the lab's current large project, and her publications. And if you haven't heard of her before, I highly suggest just looking at her TED Talk there. I said, watch me in this slide with the link. And she does tons of outreach and public speaking events. And her TED Talk's a really good place to start with her thought and the philosophy of our lab and all that good stuff. Even better, I can take a moment to show her upcoming book called Finding the Mother Tree, which is coming out in 2020 by Penguin Random House. And it's going to be part biography, part science. It should be a really fun read that kind of encapsulates her experiences as a as a human being and a professor and a mother and scientist, with also getting into some of the science behind what we're doing. So here's a brief little introductory slide. So what are mycorrhiza and what do they do? So mycorrhiza, they're symbiotic structures that are formed between a plant and a fungus. They happen underground, and what happens is that the plant gives the fungus sugar, which the plant has a lot of, and plants are really good at making sugar from photosynthesis. And the fungus, in exchange for the sugar, it gives the plant nutrients, which is from the soil. So fungi are experts at two main things: 3D exploration with their hyphae. So they're network beings that, when they're not a mushroom, they're threads. And so these thin little threads can explore space very efficiently, much more efficiently than plant roots can, because plant roots are larger. So the fungus is good at exploring space, and then when it finds food underground, nutrients, it's very good at exuding enzymes and other chemicals to break those down. So they have this relationship where each gives to the other what they're good at obtaining. So there are many different types of mycorrhiza. They've historically been defined by the physical shape of the interface between the fungal hyphae, so the threads, and the root itself. Sometimes the fungus wraps completely around the root, almost like a cocoon, and mediates pretty much all exchanges that the root has with the soil environment. There's of course many species of plants in the world, right? And there's many species of fungus that do these things. And so if you take the many species of plants times the many species of fungus, there's many, many more species pairs. And some fungi are generalists in that they pair with many species of plants, and some will be specialists in that they pair with only one or two. And functionally, this is really important. As much as 70% of the plant's nutrients can come from fungi. And actually some plants like orchids and blueberries and other things, 100% of the plant's nutrients come from the fungi. And in exchange, as much as 100% of the fungal sugars can come from the plant. So usually it's less, but for certain species pairs, this is obligate, so necessary to have these partners. Otherwise, neither of them can grow. So on top of this is this idea of common mycorrhizal networks. So this is a major focus of Suzanne and our lab. And this is when one fungi has this mycorrhizal relationship with multiple trees. And so you can see in this picture, this red ectomycorrhizal fungi. So this is the one that wraps around the outside of the root. So ecto meaning outside. And it partners, as you can see, both with this beech tree and the spruce on the right. And it connects them together. So what can happen here is that the plant will pass to the fungus carbon, so that's on the bottom, and carbon usually in these cases comes in the form of sugars from photosynthesis. And carbon, nutrients, water, the tree can pass this to the fungus, and then what happens is we find that in the other tree. And so in a sense, this fungus is an intermediary between the trees and makes what we call the mycorrhizal network. And water, nutrients, and even information is shared. So we've shown in our lab jasminate, as well as many other chemical molecules, are passed back and forth, and plant hormones. So these are really important, and we're kind of just at the beginning as scientists in understanding what these do, how they do it, and how plants communicate through this network. And intriguingly, these networks exist even across species, and plants will help each other across species lines. So in this picture, for example, the beech, which is very efficient, of course, broadleaf trees, they don't have their leaves in the winter, right? So in the early spring, what happens is carbon flows from the spruce to the beech tree. And then later, after leaf out in the summer, when the leaves on the broadleaf trees are growing and productive and doing things, photosynthesizing more efficiently than conifers do. The flow reverses and carbon moves back to the spruce, only to reverse again in the fall. So in this way, they kind of even out each other's carbon production. So very briefly, one of the big projects that's going on right now in the lab is called the Mother Tree Project. And this is a photo of Lonely Doug, which is a giant Douglas fir on Vancouver Island. And the Mother Tree project focuses on the legacies of these large trees, which we're calling mother trees. And the reason we call them mother trees is that underground, you can imagine Lonely Doug's root system here is massive. And it's connecting to all of the new young trees that are growing up. And these sharing resources through the mycorrhizal network, especially water, information, and carbon, Lonely Doug actually feeds the trees that are growing up underneath of it. So usually, of course, we don't think of these things when we're clear cutting forests, right? The big trees are the profitable ones. So the Mother Tree Project is a large-scale experiment across BC. There's a little map I put in the lower right corner of the slide. So it goes from southern BC to northern BC, mostly in the interior. And it covers a large geographic distance to follow a climate gradient. So that's the proxy for climate change. And it evaluates the effects of mother trees on what we call regeneration, so young trees growing up, and a slew of other things. So what we call variable retention harvesting. So how many of these trees, if we leave them on the landscape, how much does it affect sort of regrowth after clear cut? Species mixture, carbon cycling, soil microbes, fungi, of course, applying different seed stock when we're replanting, wildlife, and on and on. It's a massive project that's underway. Just to give you an idea, this is the scope of the ongoing project. So Suzanne, I think she told me she's had over 100 students over the years. So give you an idea of the diversity of the things we're working on is Dr. Teresa Ryan. So she's working with me on the Salmon Forest Project. And she studies salmon, orcas, especially Aboriginal fish traps and alternative harvesting methods. There's myself, of course, I'll get to in a moment. Wamani, he studies also in the Salmon Forest Project, and he studies indigenous perspectives on mother trees. Dylan's a new addition to our lab. He studies the soil of Haida Gwaii, which is a very large island. I think it's the size of England off the coast of BC and Alaska. Laura Super studies roots and shoot microbiomes. Amanda Assay studies kin selection in mycorrhizal networks. So she's focusing on, in that picture of Lonely Doug, feeding the young trees that are growing. She studies, do trees feed preferentially their own children? And it turns out they do, and her results are super interesting. Dr. Camille Defend, she just graduated from our lab. She studies roots and architecture in regards to climate change. So how, of course, are trees responding to changing climates? Are the roots moving deeper when it's drier? And to what degree do they have the capacity to do that? So how plastic are their responses? Katie, who we'll hear from after this. Alexia, she studies wildlife in the Mother Tree Project. Elena Evans, soil reclamation from tar sands projects in Alberta. And she's recently graduated, is now running an urban farm called City Beat Farm that I had a box from last year, and it was delicious. Delicious vegetables. Dixie, she studies armillaria root rot. So again, not all of these fungal partners are necessarily benign, right? So armillaria is a very interesting case where it has a mycorrhizal symbiotic relationship with the plant most of the time. But if the plant gets sick or there's a drought and it starts dying, armillaria will switch and start eating the tree and finish the job. So that's a very interesting case. And Gabrielle, who studies logs and what we call coarse woody debris on the forest floor, and how mycorrhizal networks contribute to degradation of these types of wood. So a huge group of people, there's tons of people I'm leaving out of this. And I just personally feel really blessed to be working with such a talented and keen group of people. So despite that diversity, I just want to touch on some of the themes that connect us as a group. And that number one for me is complex systems and complexity thinking and theory. So how do we approach things as whole units rather than breaking them down into parts? And to what degree can we break down things into parts before they lose their essential adaptive qualities that interest us as both managers and ecologists? Part of that is what we call adaptive management, which is a system that involves observing and listening to nature, right? Are you fishing too much fish? Well, you need to continue watching the fish populations continuously, right? To see how they're doing. That, and change what you do as a manager. Soils. The soils are probably the most complex ecosystem on Earth. They're dark, they're deep, they're three-dimensional. They have air, water, earth, all in one, life throughout it. And we're just beginning to understand how soils work, which for me personally is fascinating. As an agricultural species, soil still is chock full of mysteries. It's really one of the frontiers of science. And then connection and community. So connection between organisms, between humans and the environment, individuals and the land, where home is, right? Which is very important, I think, for gardeners and stewards in urban environments and farms. And our connections between each other too. So how can we look at all these things and manage all these things in fair ways and efficient, productive ways that is good, not just for us, but also for nature and the animals and plants that we share this world with. So I'm going to move on and get into my work. So very briefly, it's called Fish Forests and Fungi, Mycorrhizal Networks in Salmon Forests. And I have a slew of donors here, the Donor Canadian Foundation, the Brinkman Group, TerraWeb, MyTex, UBC. Research like this always involves kind of constructing these networks of people to pay for it. And it's usually really hard, and it's hard to get money, and it's hard to build coalitions to get work done, especially for 4 or 5 year projects, which is the length of an average PhD. So very briefly, I'm going to talk a little bit about myself. So this is the city I grew up in, Toronto, Ontario, which is one of the great cities of Eastern North America on Lake Ontario here, and current NBA world champions. My apologies to the Bay Area, but people here in Canada are so stoked about this. And you can see in this picture, it's the biggest city in Canada. Millions of people. I think there's 8 million people living in what we call the GTA, the Greater Toronto Area. And yet you can see how green this is, well, or orange, it's the fall in this photograph. And this is a river valley near my home called the Don Valley. And the lower left, you can see just a sliver of this river. I don't know if you can see my mouse cursor, but over here. And of course, this valley is dominated by these highways, right? These arteries of pollution pumping cars through the forests. And as a kid, I hated cars and I hated highways because they really chop this valley up into fragments and reduce access. You can't walk across a highway, right? So we have this beautiful area and there's only a couple entranceways to get into it. And so I grew up playing in these valleys, mountain biking in these valleys. And even though I was in, a great North American city, I still grew up strongly attached to nature and strongly attached to this idea of nature and natural spaces and the city as my home, right? So that, as I grew up, and that brought forward in my mind many questions, right? How does the city relate to nature? You know, the trees in our yards, the gardens, you can see in this photo on the right, that's a neighborhood, right? There are houses under those trees, but from the air, it looks like an urban, well, it is an urban forest, right? So what is our relationship with these forests, both psychologically and also materially, right? How does it change water flow? How does it change nutrient flow, air quality, all this kind of stuff? Where does sewage go? And other stories. So, of course, again, taking on this watershed perspective, all our sewage in Toronto goes straight into Lake Ontario. And where does our drinking water come from? Lake Ontario, right? When as a young child, this was very immediately impactful to me. And for when I became older and taught kids and young adults, these questions, if you're a teacher, taking your kids to a sewage treatment plant is really an eye-opening experience. What I call the magic of brown spaces. So these are the forgotten spaces, right? The industrial factories that have been abandoned, these kind of things. So they're the wild spaces in the city. And I was a big geek growing up, and so what I call wood elf versus orc mentalities. So if you know Lord of the Rings, right? The orcs are this kind of industrial, exploitative, settler colonial force. And contrasting that point of view with, the wood elves who live in harmony with nature. And furthermore, as a vision, adding to nature. So not being a negative force, but a positive force on the earth. And in science fiction, I've always been obsessed with what we call garden worlds. So imagining our planet as a garden, right? So Toronto, incidentally, this river valley has been ignored and largely fallen into disuse. But finally, the city may be ready to proceed on improving and taking care of the national treasure. And so there's a link there to an article about that. So diving forward, for a city kid, I was interested in all these ideas, interested in mathematics, interested in complexity and complexity theory and how all these things tied together, and happened to find myself in this place. So this is outside of Bella Bella in Haldsik Nation territory in the Great Bear Rainforest. And here's a map. So it's north of Vancouver, about halfway to Alaska. And so here in BC, we call it the Central Coast. I put LA in there for a sense of distance. And it's a rainforest and temperate rainforest and not too different from Washington and Oregon. So what I was doing there was I was in these beautiful small streams and we were studying this bird. This is the Pacific wren, and I highly recommend listening to its call. It's very beautiful. And I was studying this wren along streams with different numbers of salmon. And so the question was, the salmon come, of course, every fall and to spawn. And when they spawn, they only spawn once. So they come in their hundreds and thousands and millions. They sneak their way up these tiny streams and they lay their eggs and they die. And when they die, or when they come up the streams, of course, they're attended to by bears who consume them, and they drag these corpses all throughout the forest. It's a slaughterhouse. And of course, the bears leave the corpses everywhere, and from the bears, it goes to the 10,000 things. So ravens, eagles, beetles, flies, mink and otters, zooplankton and other things in the biofilms in the streams. And so the idea with the bird project was, do the birds, if the energy goes from the salmon to the insects to the birds, how much does it change the fitness and nesting success of these little Pacific wrens? So this got me thinking, if the salmon nutrients goes into the soil and into the trees and into the insects, well, first it has to go through the soil, right? And if it goes through the soil, it has to go through the mushrooms, and maybe then this idea of mycorrhizal networks comes in, and maybe the mushrooms play an active role in distributing and capturing these nutrients, nitrogen, phosphorus, potassium, especially calcium, and passing them on to the trees. And you have to remember, this is a place where it rains 2 meters a year, buckets, just buckets. And so if these nutrients aren't captured quickly in the soil, they're just going to wash out to sea. So my approach is looking at 26 different streams along a gradient of salmon density, so 0 salmon to thousands. And I'm looking at 5 streams of waterfalls that block salmon access. So that presents within a small area, 10 or 20 meters or so, a sharp transition of density from high salmon to low salmon. And that's going to allow me, with these 2 kind of treatment groups, to examine how salmon change the soil, change the mushroom populations and the vegetation along these streams. So briefly, the approach I'm using is that there's most of the time in ecology, there's 2 important questions. What's there and what are they doing? So to answer the question, what's living there? Of course, when you're studying soil, it's hard to look at these things. It's dark, right? It's wet. Most of the things are microscopic, right? So on the right here, I have a photo of mushroom, and that white stuff on top of the moss there, that's the fungal threads. We call it hyphae. So if I look at that hyphae, I can't tell you what species it is morphologically, right? One thread is the same as another. Even if I look at this mushroom on the left here, this red russula, it's really hard, even for world experts, to tell me exactly what species that is. So what you need to do then is use DNA. To answer the question of what's living here, I'm using what we call next generation environmental sequencing, which starts with me taking a soil core, which you can see in this picture. I bring that into the lab, I extract the DNA, do a process called PCR on it, and send it away for sequencing. And what I get back is this table here, something like this, that is an OTU table, which stands for Operational Taxonomic Unit, also known as species. And the reason we don't use the word species is that in fungi, often they don't have sex, often they have when they do, they sometimes have many different genders. It's really hard to say what is a species and what is an individual. In fungi, unlike us, all our cells, for example, are all genetically identical. In a single fungus, different cells can have different genomes. So that's like multiple species living inside a single individual. So to get away from that, we use this more obscure terminology. So that answers the question of what's living there, or at least starts to. The second question, what are they doing, is I use what we call stable isotope patterns. So talking briefly about nitrogen, nitrogen comes in mainly 2 forms: normal nitrogen, which is N14, and heavy nitrogen, which is N15. So it has an extra neutron, but the same number of protons. And so what that means is it behaves the same chemically, so you can't really tell the difference between them, except one is heavier. And the reason that's interesting is you have a picture on the right here, and different things have different levels of N15 in them. So in the lower left, so lower N15 levels are on land. And you can see on the right, in the marine ecosystem, they tend to be higher, right? So especially the piscivore fishes, so fish that eat other fish, like salmon, have very high levels of N15. And so that because they're, 1, from the ocean, and 2, they're predators. Incidentally, you can measure human hair samples in the N15 levels, and you can tell if someone's a carnivore or or rather an omnivore or a vegetarian or not. So what this means is I can take samples of leaves, mushrooms, and other things in the forest, and if it has high N15 values, it means that nitrogen that's feeding those things is coming from the fish. So here's a very complicated picture, a simplified model of nutrient flow in these forests. And you can see how complicated it is. There are many stocks and many flows of nutrients in any forest, or really any ecosystem, our cities included. And so by following patterns in these N15 levels, which we can measure very easily and cheaply, we can make inferences about how nitrogen is moving around. And so that's called staple isotope analysis. That's one of the main tools we use. And on the lower right is a picture of my data. And so this is salmon density as it increases. You can see that the 15N, the heavy nitrogen in mushrooms also increase. So here's a map of where I work, and everything's boat access and very remote. And here's a picture. If this is my thesis in one photo, this is what it is. Here are the small vertebrae and ribs and fin bones of a dead decomposed salmon. And these little mushrooms, these are nitrophile mushrooms. They love nitrogen, are growing out of it. And that white gunk kind of throughout the pine needles and wrapping itself around those bones are the hyphae of this mushroom. Here's another beautiful mushroom, Anemonida. And here's another one with a little newt. So just to give you a taste of how beautiful some of these places and some of these organisms are. And of course, there's a whole pile of questions that I'm asking when I do these things. So how far into the forest does the salmon effect extend? How should this impact our management? How important is this for forest plants, fungi, soils? And how important is the mycorrhizal network in distributing those resources, which are very kind of randomly assorted both in space and rarely in time, right? They're seasonal. And finally, to get back to this mother tree idea, how important are mother trees and legacy effects in management context in these kind of riparian zones? So I think I'll stop there. Pamela, do you want to take the mic back?
Yeah, I will, and introduce Katie. So Katie, I mean, that is just, my head went on some of those ideas. I look forward to getting to the Q&A. But Katie, do you want to get on? So Alan, can you hit unshare?
Oh yeah, I gotta do that. Hold on.
And I think that what is so interesting about all this discussion of mycorrhizal networks is just how many different applications there are to be thinking about them. I mean, it's pretty incredible.
So sorry. Oh, stop share. Aha.
There you go. Okay, great. All right, Katie, if you will go ahead and share your screen, we are in business.
Great. So first off, thanks for having us, Pamela. It's been really interesting learning about the long history of the Pacific Horticulture Society, and I'm excited to share a little bit of my research today. So before diving in, I worked for 5 years in the mining industry doing mine reclamation work. But it was my meeting with Suzanne that really got me interested into below ground ecosystems and their complexity and really their importance. And so my research today is looking at recovery of below ground ecosystems and mine reclamation. Yeah, so I'll dive in. First off, I don't think I really need to do a lot of convincing with Pacific Horticulture Society group, but just a brief history or overview of the importance of soil organisms. So from a physical perspective, they're ecosystem engineers. They're tuttling, they're mixing, they're forming all these pores and giving the soil structure. Even microbes are using their hyphae to, the fungi are physically binding soil together or secreting these glues. And so all of those biological processes help build soil structure. In terms of nutrients, it's really the soil organisms that take the plant litter that you see here and turn it into sources of nutrients that are actually available for the plants to use. And then finally, soil organisms, of course, carry out these mutualistic symbioses that we've been talking about. So a great example is nitrogen fixation, which provides most of the nitrogen in our soils. And then finally, mycorrhizae, which Alan has done a great intro on and is going to be a focus of my talk today. So it's all well and good to talk about these really beneficial soil organisms, but oftentimes we're not so fortunate to be working with soils that have a lot of them. So this is a photo from my field site at a mine with just some overburden that is being used in reclamation. And you can tell from the photo that it's not exactly high quality soil. And maybe you've had experiences in your own garden or in urban environments where in a similar way you're dealing with soil that is poor physical structure, not much in the way of nutrients, organic matter, and in fact really almost has no biological community. And so what my research is looking at is, well, what are techniques that we can use to help recover this soil food web? Recognizing that these soil organisms can help seedlings establish in these really harsh environments and give them that resilience, and that these organisms will carry out the process of over time building and improving the soil. And so to do this, the central concept that I'm working with is called ecosystem legacies. So if you think of a disturbance on the landscape, there are always little patches of the ecosystem that survive, whether it be seeds or spores or the mother trees. These are all little pieces of memory, ecosystem memory, that can be used to help the system rebuild itself. And so what we're trying to do in the first part of my research is using soil itself as a type of ecosystem memory. And so I'm working in a remote area near a forest at a mine site, working on a reclamation project there. And what we're doing is we're taking small volumes of healthy forest soil and mixing it into the rooting zone when we plant a seedling. And so the idea is that we're inoculating it with all these beneficial microbes that are native to the area. And we're also inoculating it with the full community of microbes. So you can certainly get commercial inoculums of mycorrhizal fungi, but it might be 1 species or 3 species. Here you're getting numerous species, as well as all the bacteria and other microbes that they actually work together with to carry out their functions. And so this would be a method that's applicable in an area where you're working with really degraded soils or you don't have any topsoil available. And then in addition to getting this biological inoculum, you might also get a little improved rooting zone in this seedling where they can sort of get standing on their own 2 feet from the extra kick of nutrients and improve soil conditions. So to test this, we did an experiment in the greenhouse at UBC. And so I'm going to show some results from willow seedlings. And I'll walk you through this graph here. On the y-axis is basically above ground biomass. And across the bottom are different amounts of forest soil that we added to seedlings. So on the left, we've got the control, which is just forest soil. And then over here, you see overburden, which is basically that pile of soil I showed you earlier. And you can see when we added different volumes, this was equivalent to about 50 mils of soil mixed in and about 200 mils of soil mixed in. We can really see the improvement in growth. And then the third section here is the same thing, but in mine tailings. So mine tailings with no forest soil, mine tailings with a small amount, and mine tailings with a little bit more. And so interestingly, with these seedlings, we harvest them in the greenhouse and wash their roots so we can look at them under a microscope. And from that, we can see what percentage of the roots are colonized by mycorrhizal fungi. And so you can see here that the pattern is really the same as we're seeing in the plant growth. So the plants are growing better, and they're also colonized by more fungi with these transplants. So that's showing that they're doing what we were hoping, basically. And so I interestingly did a comparison where I did the same experiment, but I sterilized the soil, and so there was no living matter. And that really was kind of proof of concept that the biological component of the soil is really important for providing this growth benefit. And so thinking about this concept of ecosystem legacies, it's important also to think about what kind of ecosystem you have underground. Different plants create and use different below ground ecosystems. So and you can show this through the type of mycorrhiza that they associate with. So on the one side here, we have ectomycorrhizal fungi, and they're really visible on the root. They form kind of a coating on the root and change how it grows. And so they mostly form with conifers and coniferous forests. And then on the right, we have arbuscular mycorrhizae, which are also symbiotic, but they've evolved quite differently. And you can't see them you can't see them under a microscope. They actually form little structures inside of the root. And so they form with lots of agricultural species, herbs, tropical trees. And so you have these very two sort of different below ground groups. And so we were interested in doing a study to look at how these different groups will affect transitioning an ecosystem. So what we did is we grew cedar seedlings in a greenhouse in control soil, or soil that was conditioned by spruce trees, or soil that was conditioned by willow trees. And so you can see here in the graph, the cedar trees that grew in the control soil grew the best, whereas the cedar trees that grew in soil that previously had spruce seedlings growing in it grew not as well. And the soil that previously had willow growing in it, the cedar seedlings grew really poorly. And again, so the other side of it is showing the mycorrhizal colonization of the cedar trees. And so they form that arbuscular mycorrhizae, the one that is on the inside of the root. And so the colonization of that was the highest in the control soil where growth was best. And then the percent colonization decreased and corresponded with the lower shoot biomass. And so what's interesting about this is that cedar are those arbuscular, but spruce actually form the other kind, ectomycorrhizae. And willow form very, very weak arbuscular mycorrhizae. They don't really rely on mycorrhizae in the same way. But what that tells us is the plants that you're planting are changing the below ground ecosystem, and it doesn't always benefit every species. It's going to selectively benefit the species that rely on those same relationships. So here, in this case, we're seeing there's probably a loss of that arbuscular mycorrhizae in the system over time when there is no cedar present. So this is kind of applicable even in your own backyard if you're transitioning part of your garden to from something that used to be perhaps growing some little conifer trees to a vegetable garden. It's going to not only take time to shift the plants that you're growing, but that below ground ecosystem has to shift as well. And then the third legacy that I want to talk about today is something really relevant to mycorrhizal networks and really lines up with what Alan was speaking about earlier. And it's the concept of spatial connectivity. So when we think about disturbances on the landscape, a lot of research on this has been done in forestry where you have big clear cuts. And we hear a lot about the importance of leaving patches of trees and having this connectivity to have habitat corridors and for above ground organisms. But there's no reason that that doesn't apply to below ground organisms. So there's actually some interesting research that shows the bigger the patch of trees on the landscape, the higher the diversity of soil fungi and bacteria. And so certainly some fungi can transport their spores long distances, over 100 kilometers, but others really, they tend to spread more through root-to-root contact. And so thinking about that, these spatial connectivities on the landscape can really help maintain and share that biodiversity below ground. And there's quite interesting research showing when you leave these patches in a forestry context, the seedlings that are growing 10 to 20 meters away actually have a more diverse set of mycorrhizae colonizing them, and they are forming ectomycorrhizal networks with those remaining trees. So those trees can be giving them carbon and various resources. And sort of a really good way to summarize that is somebody did a study and found that in 75% of the cases, trees that are connected to these mycorrhizal networks are benefiting and receiving resources. So I wanted to look at this in a mine reclamation context where you've got a really, really disturbed zone up against a fairly undisturbed forest. And so this is one of my research sites here, and what I've done is I've planted rows of spruce seedlings out from the forest. And some of the groups are sort of connected to the forest, whereas others have these large trenches excavated, which separate them from the below ground. They separate below ground connectivity with the forest. And so I'm looking at how soil fungi, bacteria, and even larger fauna, things like mites and beetles and spiders, are ingressing, and how that can relate to improved vegetation recovery. And so this kind of concept, I think, it's not only applicable in these industrial settings, it's also good to think about in urban settings. You know, how can we preserve these intact or old little patches of ecosystems? And how can we plant groups, or how can we plant species in groups that will work well together and have compatible mycorrhizae to form mycorrhizal networks? And so with that, I just wanted to give a quick thank you to Suzanne and my supervisory committee. And of course, I don't do all this work alone. There's a team of us in the Below Ground Ecosystem Group, and I work with lots of research assistants and volunteers. And then of course, a number of funders have made this work possible. Thank you.
Super. Okay. Okay, so that is very interesting. Thank you, Katie. Thank you, Alan. You've taken us into a world that most of us don't think about, but is actually, I think, really applicable in urban settings, where we are fighting against pretty degraded situations. You know, one of the things that I was thinking about is we have a lot of fear around degraded sites. This is mostly on large scale situations where, let's say, we're building new, you know, elementary schools or natural history museum. In fact, in Los Angeles, this was an issue in building the Natural History Museum on a site that had degraded soil and pollution in the soil. And the remedy was scrape away as much of the soil as you possibly can, and then bring in fill, but not really a concentration on what that life is below ground. And so I'm wondering, you know, do you think that the research that you're doing can start to inform us about practices that we might want to begin using to reclaim these degraded sites without having to, you know, absolutely destroy all the soil that is on site and remove it?
Yeah, I mean, I think there's certainly recognition more and more of the issues of moving contaminated soils. I mean, to start with, it's expensive. You're emitting a bunch of greenhouse gases by trucking it out, and you're really just moving the risks of that contamination to a new location. And so I'm not a contaminated sites expert, but there's this whole body of research on biological treatment methods where you use the microbes to break down or alter contaminants to a less harmful state. And thinking about mycorrhizae, there's research showing that they can actually help plants be more resilient to contamination. They can provide sort of a physical protection on the roots, or even they themselves can break down contaminants. I mean, it could really be the subject of a whole nother webinar, but I would say in general, these options aren't being as widely implemented as they could be, whether the science isn't there or there's a bit more of a sort of a knowing doing gap. But I think you do see more and more people trying to get creative management approaches. I know in Vancouver, there are a number of examples where there is some soil contamination, and maybe you wouldn't want to have a playground there or grow vegetables, but it's fine to be a street side garden. So looking at ways to just be able to leave that soil in place and have it be safe, but allow some of the biological processes to continue.
Yeah, we're going to have to figure that out, because carting soil around is definitely going to get more and more difficult.
I'd add there's tremendous potential to combine the doing with with the science, right? We have all these soils around, and you know, we should just start doing stuff and get people looking at what works and what doesn't work, and later you can start asking why, right? But
Yeah, that's a really good point. I think another question that I have that's kind of fits into this is, so you all are in British Columbia and Pacific Northwest, you know, working on forests, and there's a lot of biomass, and, you know, it's super verdant, and as Alan said earlier, rains buckets where he's doing his work. Let's think about the whole Pacific region. You know, in Southern California, we can't just go to a forest and grab soil out of a forest. And would that even be appropriate, you know, in a landscape that is not receiving buckets of water? What do you think?
Yeah, I mean, I would say that it's definitely not necessarily something that's applicable to urban areas. And when we're talking about doing these soil transplants at somewhere like a remote mine site, we're not promoting going and digging up, you know, park soil from down the road. But I think it does help develop the concept more as protecting urban soils as a resource, and trying to, if an area is, for example, it is going to be disturbed, making sure that soil is saved and making sure that it's used somewhere else as soon as possible. I mean, the longer that it sits, the vegetation, the seeds and propagules are going to die off, the microbes are going to die off. So just use it right away and use it somewhere. Or maybe even in your own yard, if you have a really happy vegetable garden that you're planning to expand, maybe you can mix some of that soil over as you're extending it. So I think it's, I mean, to use a cliche, we're just trying to add another tool to the toolbox. And in urban areas, you can maybe it's better to look at some of the other alternatives like compost teas.
What about community-wide, going to places that do already have native gardens, let's say, that are well established, and sharing, you know, asking them to share some of their soil? And like, how do we get it? I mean, do you just like scrape it off the top, or do you dig? I mean, can you give us just a little insight into how that would work?
Sure. I mean, the most biologically active part of the soil is the very top layers. It's where there's most of the organic matter is concentrated. It's where a lot of the biota hang out. It's where a lot of the tree roots concentrate to get nutrients. And so that's really kind of the gold there. The deeper you go, the fewer soil organisms, basically. But just when you were saying that earlier, something that crossed my mind is it something that you could maybe work on promoting at the nursery stage too? If you're growing things in a nursery and growing them in, keeping that soil alive, starting from square one and transitioning those seedlings into your own garden, that in itself could be sort of an avenue to explore.
yeah, and I think, you know, something gardeners already do all the time is trading plants, right? And when you trade the plant, you keep soil attached to the roots. And you know that in my garden, which I'm just starting, it's the transplants I've gotten from friends and family that are really kind of doing the best. And you know, it could be a part of that. They're bringing with them their communities, right? Already on the roots.
Yeah. Well, I know a little bit about the nursery trade. Has been kind of going through a revolution over the last decade, at least, maybe longer. You know, whether or not to apply any kind of inoculant to the roots of plants in the cans, you know, before they're sold out to people. And that's been a, I think, ongoing thing. I don't know how it's resolved, but maybe that's a topic that we should take on, get some nursery people in talking about to inoculate or not, and what the results are doing that. You know, something else that both you, Alan, and Katie touched on was this idea that the interconnectedness of all things, and especially when we talk about gardens, that, teasing everything's apart is very challenging and fruitless, actually, quite actually. Um, but is there maybe a way for us to begin to think about, the biodiversity projects that are happening that are trying to preserve sites and then also create new garden landscapes that promote biodiversity? Can we just talk about the interconnectedness of things and just how maybe that's a symbiotic relationship between planting for biodiversity and then also creating more biodiverse soils? That these things kind of go together and we need to be thinking about that.
Yeah, I think they go hand in hand, right? I mean, gardeners know just through trial and error, for example, like Katie's results with cedar and other plants, gardeners know certain plants grow well together. Companion plants, there's such a thing as companion plants. And still don't know why exactly, but almost certainly it's the soil and soil underground story is what's going into it. And so in cities, one of the things that fascinates me about urban forests and urban gardens and spaces is that frequently they're hyper diverse, right? When I walk into that forest that I showed you pictures from, I can know every tree, and there's only 10 species of trees. But when I walk down my street in Vancouver here, there's hundreds of trees from all around the world, native or, usually some, from Southeast Asia or wherever. And that's, I think, really interesting because it's just kind of this mixing pot I would keep smashing things into. And, I'm a huge fan of local planting, but in terms of diversity and how bird life and insects and other things use the landscape, for them, it looks very different than natural forests because it's more diverse.
That's interesting.
Which we don't think of cities that way, right? We think of them as deserts, and they can be. So who knows what's going on underground, right?
Yeah. I mean, it's possible that, do you think that these really super diverse forests are performing better together, or that this is maybe a challenge for them because they're not growing with plants that they're used to growing with? And in terms of biodiversity above ground, they are deserts because many of our local species are so closely paired to particular plants that when they don't exist or are, there are a lot of non-natives, that is equivalent to being a desert for the insects and birds that rely on those local plants.
Yeah, for sure. And everything's like, we're in the natural forest I work, there's a cadence to the season, right? Every season has its own berry is the cliche, but it's true, right? And every season, I have a colleague up there, she studies along these salmon streams flowers, and are there more flowers and brighter flowers with more nectar, for example, along these salmon streams, because the plants of the flowers have more access to more nutrients and energy. And one of the things she's finding, of course, is that there's a cadence to what blooms when, and there's a certain sequence that there's usually something blooming on a normal year, right? Of course, there's more and more non-normal years. Right? So sometimes if something's late, then the bees find themselves in trouble because
Yeah, that just building on what Alan was saying, it really sort of that critical need for us to have more resiliency in our ecosystems. I think thinking about mycorrhizal networks is a piece of that. There's a lot of research just showing that connectivity can help trees be more resilient to just various stressors, be it drought, pest pest attacks. And so I think urban areas can be especially prone to some pretty intense conditions for plants. You know, the hydrological cycle's been really messed up. They can get really hot. And so allowing our plants to grow in these more diverse communities and create these networks, I think is a route to explore for resiliency, especially in this changing climate.
Yeah, and I think that really highlights, it's not, you know, we talk about biodiversity, but it's not just diversity. Like, we do need to think about what kind of diversity. I think that's what you're talking about, Pamela, like native species especially, but also increasingly, here's an example in Vancouver and the BC coast more generally, cedars are not doing well. Just down the street, there's a big cedar that's dying, actually, on a hillside park. What's doing great, though, the last 20, 30, 80 years are the redwoods, right? Which, of course, are from down in California and aren't exactly native here. But, you know, climate change projects that we will be prime redwood growing conditions in the future. So, you know, as these things shift around, I think the questions as to also what is native kind of almost has to change because the conditions are changing. And, you know, the native communities are tremendously resilient. It can be tremendously resilient. We can help them, but at the same time, they will change, right? And there's tremendous political resistance to that idea. And I think that's deserved, right? But, you know, opening our mind to having urban, especially in urban contexts, things that will grow well in the very, you know, difficult conditions, you know, air pollution, soil compaction, all these things, I think is really important.
So I wonder, you know, and this, we can wrap it with this, it kind of pulls all this together for gardeners. You know, few of us are planting forests. I mean, some of us are lucky enough to be able to do that, but most of us are planting landscapes that have a wide range of plants in them, from, you know, forbs and little grassy stuff all the way up to, you know, the evergreens and, you know, large deciduous trees. And we're wanting it all to kind of work together. Right? You go. Any thoughts about the wisdom of these, you know, of these plantings, and or maybe the not so much the wisdom of them, but the idea that you know, maybe we think very carefully about the way that we're grouping plants, even within a landscape context. Anybody want to tackle that?
I mean, I'll take a first go at it. What immediately jumps to mind for me, which is kind of a parallel with what I do in mine reclamation, is we always talk about working with the successional trajectory. And so if you have, you know, say you buy a house with a big yard somewhere in Vancouver, and you want to grow a nice, you know, mini old growth forest, you're not going to get there right away. The conditions haven't been created. And so even if that's your long-term goal, you sometimes still have to think about in the context of tree years, not human years. And so maybe you want to start working with some of the native species, but the shrubs like alder in our case that are more resilient in these early successional conditions, and over time they'll build and create the soil and sort of microclimate environment that the things you ultimately want to grow will survive in.
You mean not instant gardening? Well, in Southern California, we're never going to have that kind of forest. Our big objective would be having some kind of an oak woodland.
Right.
Savannah or something. And yeah, I think my thought on that, Pamela, is this adaptive management perspective, right, that we're saying as gardeners, observe your garden, right? See what does well where and why and with which other plants, and kind of keep tinkering at it, right? Science you know, we come to these big general conclusions, but they're not necessarily applied to your little plot of land, right? And only you can know that, and you can use science to learn about it. But really, it's about, you know, for me, that's the art of gardening, right? Is really paying attention and listening. The other big thing that I'm trying to apply in my garden is one of the stories, of course, the big story, one of them in the world these days is the collapse of insect populations. So insects in Europe and Puerto Rico, we know for sure, are down 70% in the last 30 years. It's catastrophic, right? We don't talk about this enough. And so trying to manage our lawns, especially as insect habitat, is I think really important.
What's a lawn?
Right?
Right. Yeah, I think as you both have said, it's the interconnectivity of all things, and you've opened up a fascinating world for us. You've started a conversation. I expect that we will have more dialogue about this over the coming months and years. And I want to thank you both very, very much for taking the time to talk to us today and to put together these presentations and also to bring it into context of gardening. If you will bear with me for 1 second, I am going to, maybe I'm not. What did I just do? Oh, yeah, sorry. Those are my notes. Here we go. Okay, there you go. Oh, I shared my desktop. That's what I did. Oh, well, you'll bear with me. Okay. So, well, Alan, you're going to be with us in this. So just smile. You're on my desktop, and I can't figure out why. Anyway, thank you, everybody, for joining us for this edition of the Digital Classroom. We are, there we go. We have several articles from the archive of Pacific Horticulture that are worth taking a look at. The first one from fall 2015 is called Tending the Urban Forest by Jeff Reimer. I think that's how you pronounce it, and forgive me if it's not. That whole issue has a lot of information about trees in it and is particularly interesting and pertinent to this conversation today. Also, we have an article from fall 2003, Horticulture Beyond the Backyard, A Tale of Three Ditches by Deborah Jones. Now, this is very interesting because this talks about, in the Pacific Northwest, what observing gardening with salmon runs in your backyard. And I think is quite interesting to see what kind of plant material is best used in those situations. And then finally, we have an older article from fall 1991 called Why Trees by Russell Beatty. And this is a fabulous article about the conclusion that we have to have trees, especially in urban settings, but not for just the reasons that you can think of off the top of your head, that there are greater issues at hand having to do with community and so forth. So those are great articles, but I would be remiss in my job as the executive director if I did not encourage you to come to our website, pacifichorticulture.org, pachort.org, and make a donation to Pacific Horticulture. We want to continue doing these kinds of talks, and the only way we can do that is with your support. So please come take a look at our archives and also become a member and a donor. And then finally, if you have any questions about this talk or any of our talks, please email us at digitalclassroom@pachort.org. Again, I want to thank everybody. I'm coming back to y'all. Thank you again, and we'll see you around. Bye.